Okay, Bio 30, I'm going to continue lecture on monohybrid crosses today. I just want you to know that the slides for Mendelian genetics go from slides 74 to slides 110. So that's what you're responsible for Mendel. The other piece about what I was talking about in lecture 11, so we're going to start with sex-linked inheritance. Anything that doesn't involve sex chromosomes are autosomal traits. All right, so we have X-linked traits and autosomal traits. I'm going to talk about pedigrees. I'm going to do a bunch of examples here, and I'm going to finish up monohybrid cross. So this is quite a bit of information today. But some traits are X-linked, so they exist on the X and Y chromosomes. They don't exist on the autosomal chromosomes, so 1 through 22. These are on chromosome pair 23. So we have sex-linked traits with monohybrid crosses. So some common examples of monohybrid traits that are X-linked: red, green color blindness. There's a couple other examples that I'm going to discuss. One thing that we'll also talk about is barr bodies, and so when we look at female chromosomes, right? There's two X chromosomes. This does things like determine coat color in different types of animals. This is a calico cat. Calicos are always female. Recall that epigenetically, you can have one set of alleles expressed and another one raveled up. That's what barr bodies are. So one X chromosome is active and the other one is not expressed. So which part of the X chromosome or which X chromosome expressed is random. And the coloration pattern in calico cats is determined by that because color is on the X chromosomes in cats. I'll talk about multiple alleles after I talk about X-linked traits. X-linked McGraw-Hill, page 603. So if we look at X-linked traits, this is one example that we'll need to do. So this is page 603, McGraw-Hill, questions 8 through 10. I'm going to address this. So this is page 603, numbers 8 through 10. McGraw-Hill. Okay, so question number 8 says, a woman who has normal vision in the heterozygous genotype X big C, X little c, marries a man who is colorblind. So with X-linked traits, so X big C is normal, and X little c is colorblind. So here's the deal with X-linked, right? Males are disproportionately affected because they only have one X chromosome. So they can't have a dominant X to cover up a recessive trait. So they'll just possess the trait. The question says, what is the expected ratio of genotypes and phenotypes among their children? So these are the breeding pairs. So you make a Punnett square just like you would with anything else. Okay, so this is what you get, right? So the Y chromosome gets delivered over. Now there's a couple ways that this question could be answered. One is how many children would be expected to be colorblind? And I should rephrase that. What is the probability a child would be expected to be colorblind? Okay, so with X-linked traits, you can divide by gender or not. So this question doesn't specify by gender, so you would look at which ones would give you colorblind genotypes. So this and this. So you would expect 50% of children to be colorblind. Okay. Another way this could be asked is how many, what is the probability of children to be normal? Right, so this would be that would be a non-colorblind female, and that would be a non-colorblind male. So 50% of them would be expected to be normal. Again, I'm not specifying on gender. I could ask you another one. What is the probability? So now I am starting to specify by gender. What is the probability that the offspring is a carrier? Okay, so a carrier with X-linked traits is a female because they have two X chromosomes, and the female is a carrier. The female does not possess the trait but carries it on to the next generation. So I'm asking how many are that? So you would expect 25% of the offspring to be a carrier. Okay, another way to ask this is if male, what is the probability the offspring is colorblind? Okay, so I would only look at the males, and so if we know it's a boy, I only need to look at XY. So 50% of the boys should be colorblind, and 50% should be normal. So 50% boys, half of the boys should be colorblind, and you're generally not going to see this. Where it's going to end up being the same probability for males and females. Usually males will have a higher probability. But this is a female carrier with a colorblind male, so that's why there's the difference. And then 5 is if female. What's the probability they're colorblind? And you would just look at the females for that. Right? So here, the 50% of all the females would be colorblind. Okay. So you can specify by gender or not with X-linked traits. So that's an important concept to make sure that you understand. Okay, 9. This looks at fruit flies. So this shows you here, figure 17.17. White is X little r. That's considered non-wild type. And red is wild type, so wild flies will look like that, X big R. And the eye color in fruit flies is X-linked. So it says, suppose that you have one wild type female fly. So she's actually this, X little r, and one white-eyed male fly. So X little r, y. What steps would you follow to produce a white-eyed female fly? Illustrate your steps with Punnett squares. Sorry, you have one wild-type female fly. So she has red eyes, and this is a white-eyed male. How do we get a white-eyed female fly? So what types of parental generation should we have? So this is our P. All right. That's dad, and that is mom. So let's breed these together. So with this breeding pair, the question is asking, how do you get white female flies? All right, so this is your F1. Okay, so with the wild type female, you get all wild type males. And then you get female carriers. So here's the deal with an F2, right? You want to get white females. So X little r, X little r. How are we going to get that? That's what the question is asking. So I can't take two F1s and breed them together, because if I have a wild type male with a female carrier, I'm not going to make white-eyed females, right? There's not enough little r alleles. So this is kind of gross, but what we'll do is we'll actually take dad and cross him with his female offspring. So you're going to back cross a P generation with an F1, right? So you got to be Mendel is a puzzle, and you got to start to put it together. So we're going to take a P generation male, and we're going to back cross it with his offspring, and it has to be a female carrier. So that's gross, but that's what we need to do. And then what you're going to get is 25% of that F2 being white females. Okay. So that's question 9 in your McGraw-Hill. Number 10 says, in a species of dog, a mutant gene that causes deafness is found on the Y chromosome. Draw a Punnett square to show the outcomes of a cross between a male dog whose father is deaf and a female dog whose father is not deaf. So this is question 10. So female, it doesn't really matter what the female is, but dad is deaf, right? So big D equals not deaf, and little d equals deaf. So that is A. So this is kind of weird, because the chromosome, the trait is actually not on the X, so we don't need to specify what's going on the X. You have deaf or not. So you have this, right? So you have X, Y, D, and then XX. So all of the deaf males' children will be deaf. All males are deaf. Because if you get it on the Y and you're a dude, then you're deaf. All right. So we have a male dog whose father is deaf and a female dog whose father is not deaf. And again, a female dog whose father is deaf, it doesn't matter. She's not going to get a Y chromosome from dad. So she's still going to be XX. And then you're going to have a male dog whose father is not deaf. So he's going to get the non-deaf allele from his dad. And instead, you're going to have the same thing, right? But you're going to have X, YD, and XYD. So no deaf males with this one. So if dad's deaf, then, and it's on the Y chromosome, then the male offspring will be deaf, but the females aren't affected by that. Okay, so those are X-linked traits. I just want to see what's in your key here. Okay, so here's another good example of X-linked. Hemophilia is a very common X-linked trait as well. It's on the X chromosome. So this is in your key, page 101, numbers 35 and numerical response 8. Okay, so this says, in humans, the allele for normal blood clotting is dominant to the allele for hemophilia little h. The trait is X-linked. A female hemophiliac marries a man who is not a hemophiliac. So this is kind of weird. So the female is a hemophiliac. It's X-linked, so it's X-linked recessive. So this would be a non-hemophiliac. That's what that X is. And that would be hemophiliac. Okay. So a female is actually a hemophiliac. And so she has to be X little h, X little h. A female carrier would be X big H, X little h. So that would be a carrier. Okay. So X little h, and the male is not a hemophiliac, and this is X, so he's X big HY. So in your key, it says a female hemophiliac marries a man who is not a hemophiliac. The row that indicates the probability of this couple having a child that is a hemophiliac and the sex of that child would be, right? So this differentiates by probability and by gender. Okay, so you put the Punnett square together. So Y chromosome always comes from dad, obviously. So this would be the Punnett square. Okay, so there's a 50% probability that, right, that the individual will be a hemophiliac and they will be male. Okay, so that is C. So a woman, numerical response 8 here says a woman who is not a hemophiliac has a father who is a hemophiliac. If this woman marries a man who is a hemophiliac, what is the probability of them having a hemophiliac son? Okay, so the woman has a father who is a hemophiliac. So if the dad is a hemophiliac, the female is going to get one X little h. So she's going to be a carrier. So this female has to be a carrier. So that's what that information tells you. Okay. The father is a if this woman marries a man who is a hemophiliac, what's the probability of them having a hemophiliac son? Okay, so this is NR8. So this is, she's this, and the male is this. Okay, so X big H, X little h, X little hy. So we have X big H, X little h. So this one specifies by gender as well. What is the probability that they have a hemophiliac son? So this is a son and this is a hemophiliac. So of all the probabilities, there's a 25% probability that they have a hemophiliac son, but a 50% probability that their child has hemophilia. Okay? So that is, right, so this is... your key, page 10, all right, so that was 101. Page 102, here's another couple questions. A high percentage of purebred dogs have genetic defects. Some of these examples follow: hip dysplasia, hereditary deafness, retinal disease, hemophilia, and X-linked disorder. It's common in Labrador retrievers. Dwarfism is also common. A healthy Labrador retriever has won several ribbons for her appearance in dog shows. She was mated with 2 healthy males that are Labrador retrievers. In the 2 litters produced, some of the offspring had hemophilia and others were normal. So she's healthy, but she's obviously carrier because it's X-linked. So this information is not necessary, so there's more information that's given. So that's a point that you should understand. If the female is bred to one of her male offspring that does not have hemophilia, so that's gross, inbreeding, then the probability of the female offspring of this cross having hemophilia is what? So the female dog is X big H, X little h, because some of her offspring had hemophilia. But she's bred to one of her offspring that does not have hemophilia. So they're asking, what's the probability that there's a hemophiliac female? Right? So none of the females, none of them will be hemophiliac. Right? Okay. Okay, this question, page 102 again. Tay-Sachs is a hereditary disease that kills 1 in 360,000 individuals in the general population, but 1 in 4,800 among the Ashkenazi Jews. The disease disrupts or halts proper formation of lysosomes and increases fat deposition around the nerve sheath. Individuals that are homozygous for the defective allele have Tay-Sachs and die at an early age. Studies suggest that heterozygous individuals have a higher chance of survival against tuberculosis. What type of inheritance is demonstrated in Tay-Sachs? Okay, so they have to be homozygous. So in order to have Tay-Sachs, they have to be recessive, and this is not a sex-linked trait, so it's autosomal recessive, right? So big T is non-Tay-Sachs. Big T, little t actually gives advantage. So you know, right, and so that's how you know it's not dominant, because if there is a dominant allele, it can shield some of the defective effects, but this little allele gives some protection in tuberculosis-stricken areas. So that's called heterozygote advantage. So that would be Tay-Sachs, so autosomal recessive. Okay. Numerical response 9 says a young couple decided to have genetic screening done to determine if they were both carriers of Tay-Sachs. If both individuals were carriers, what percentage of their offspring would be predicted to have protection from TB? So that if they're both carriers, the question is asking what percentage of the offspring would have a heterozygote advantage. So we make a Punnett square for that. So big T, little t, and then big T and little t. Figure out their offspring. You would know that 50% are hetero. So 50% have a heterozygote advantage. So your answer is 50 or 0.50. Okay. That's in your key. There are more practice questions in here as well. I'm going to leave those ones though. So I gave you lots of examples looking at X-linked and traditional monohybrid crosses. I want to look at multiple alleles now. Okay, So I'm going to look at multiple alleles, and then I want to look at pedigrees. Okay, so in some cases, right, although an individual can only contain 2 copies of a gene, there's more than just 2 alleles in a population, right? Blood typing is a good example of this. There's A blood type, O blood type, and B blood type. So even though you can only have 2 copies of an allele, it doesn't mean that there's not more present. So human blood types exist this way, right? So you can have A blood type or B blood type or AB or O blood type. Okay, so when we express blood type, blood type is expressed in this way. So let's say someone has A blood type. You could see this as AA, but generally we use this. So IA. So I equals stands for antigen. Okay, so someone who has A blood type, this is typically how we look at it. This would be homodominant for A. But someone who has A blood type could also be heterozygous. Okay, so they could be heterozygous, still have A blood type. So that's O antigen. B blood type looks like this, right? They could be IB or IB little I. So heterozygous. AB would be this. IB. Okay. And O blood type is recessive. II. Okay, so that's blood typing. So this follows autosomal characteristics related to Mendelian genetics. So this is a dominance hierarchy where we have A and B are codominant, so they're equally expressed. So if I look at a red blood cell, that's AB. A red blood cell will have A and B antigen. Okay, they're both equally expressed. If someone has A blood type, they just have A antigen. Shoot. Okay, they just have A. If someone has B blood type, they only have B antigen. Someone who has O blood type does not have an antigen, so no antigen. Okay. Okay, so here's the deal, right? This is important for the immune system. An individual, right, so the immune system recognizes the antigens, right? So if an individual needs blood, right, needs blood donations, if they have A blood type, right, someone who has A blood type can receive blood from other A's, but they can also receive blood from O blood type because there is no antigen, so it won't trigger an immune response. Someone who has B blood type can get blood from other B's or O's. AB blood type can receive blood from anyone, but can only give blood to other ABs. O blood type can give blood to anyone, but can only receive blood from other O's. So O blood type is really, really valuable in a blood bank as a result. Okay. So page 606, number 11 and 12. Question says, if a man has AB blood type and a woman has type A blood, what possible blood types could their children have? So, a man has AB, and let's give the female a heterozygote because that'll increase the choices of types of blood type. And then you make a Punnett square. So this is page 606, number 11 in McGraw-Hill. So this is autosomal, right? Blood type is not sex-linked. Right, IA, IB. So possible blood types are A, B, B, and A. Okay, O is not possible. So if they had an O blood type child, that would mean that this person would not be a parent, right? There would have had to have been another homozygous. There would have had to have been another heterozygous for O trait. Okay, so that's what this question addresses. It says a couple just brought home a new baby from the hospital. They begin to believe that the hospital switched babies and the baby they brought home is not theirs. They check the hospital records and find that the man's blood type is B, the woman's blood type is AB, and the baby's blood type is O. Could the baby be theirs? All right, so let's look at this. So mom is AB. So the baby is O blood type. Okay. So the baby is O blood type. Let's say dad is heterozygous. And mom is AB. So when we put this together, so there is no O possibility there. So mom, so that means dad ain't the dad. So mom was doing something wonky or they switched the baby at the hospital. So no O possible. Okay. So that's multiple alleles. Here's another good one. Number 17 says 4 children have the following blood types: A, B, AB, and O. Could these children have the same 2 biological parents? So let's put that together. So if you have